presented by Dr. Robin Felder, Professor and Associate Director of Clinical Chemistry and Toxicology, University of Virginia School of Medicine. I'm Susie Valdez of LabRoots, and I'll be your moderator for today's event. We're excited to bring you this educational web seminar presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars. Before we begin, I'd like to remind everyone that this presentation is interactive. We want to hear from you, so questions and comments can be submitted via the green Q&A button at the lower left of your screen. We'll answer as many questions as we have time for at the end of this presentation. Also, you're viewing this presentation in the slide window. To enlarge that window, click on the icon at the lower right-hand corner of the slide window. If you cannot hear or see this presentation properly, let us know by clicking on the support button at the top right or that Q&A button and let us know that you're having a problem. Without further ado, I'd like to now present our presenter, Dr. Robin Felder. Welcome.
And cell-based assays, and of course tissue engineering. This work was done in collaboration with Merck in North Wales, Pennsylvania, just to test the system we developed here at the University of Virginia. Second slide shows kind of an overview of the demand for cell culture across all of the life sciences. In the upper left-hand corner, you have cell culture of established cell lines, primary cells, cancer cells, stem cells. That's mostly research laboratories performing cell culture. Below that is biorepositories for cell banking, and that's important for places like the ATCC in Northern Virginia, which provides all kinds of research cells to the research community. Then in the upper right, there's drug discovery, so you need cell-based screening and cell-based assays, which are getting highly popular. And so each pharmaceutical company or contract research organization grows their own cells in very large quantities to support million compound screens every week. And then finally, therapeutics and biomanufacturing, essentially regenerative medicine, vaccine production, and whatever other kinds of therapeutic things may be useful from cells. For instance, some cell products create biologicals. Those biologicals can become pharmaceuticals, so that's the therapeutic approach. But the big one that's going to dominate this entire field is regenerative medicine, because we're all aging at a rapid pace, all the baby boomers, and replacement body parts will be necessary. Next slide shows the drivers for cell culture. And again, research and biomanufacturing. In research, the cell-based assays are becoming more popular than enzymatic assays, let's say, for drug discovery, because they want to essentially decide on an entire cell whether the pharmaceutical works or not and is toxic or not. Certainly physiological screening, high cell-based product production, so again, like the CROs that support pharma, more valuable drug leads, which you can get there faster using appropriately grown cells. And this is the main focus of my presentation, is the increased relevance of 3D biology over 2D biology. And picture 2D cell culture being the Petri dish or the T75 flask, and 3D biology being either, well, I'll show that in a minute. So the other area is biomanufacturing and tissue engineering. There's increased demand for cell-based drugs and therapeutics. Bench level production as a lead to scale up, and there are very few systems currently that scale in the 3D world from bench level to full-size production. Cell-based systems to replace egg-based production of pandemic vaccines. We need to get away from the egg production because it just takes too long, and you can get a vaccine out to market much quicker with a cell-based system, particularly automated cell-based systems. So the next slide shows cell culture trends, and the trends are in all aspects of cell culture: media, matrix, cell lines. And so in media, we're going from serum-based, because all the growth factors in serum, particularly fetal calf serum, and then we're moving to define synthetics, because we're beginning to sort out what those growth factors are in fetal calf serum, and we just essentially synthesize it and put it in the media, so it can be xeno-free and have less chance of raising antibodies in humans. the matrix is moving from 2D plastic to the various kinds of 3D surfaces, sol gels, hollow fiber. There are a number of them emerging, and each has some strengths and weaknesses. The cell lines are going from research-grade cells to increase use of primary cells, particularly in drug discovery, and these cells need to act as close to body physiology as possible. Also co-cultured cells, where you grow feeder cells and the cells of interest, and so the cells help each other make more organs, and the in vitro organs ultimately will be able to essentially generate hearts and livers and kidneys and lungs and be able to test those, which are the targets of a lot of pharmaceuticals. So basically for cell cultures, moving from basic research, high throughput screening and bioprotection into high content screening, in other words, looking for a lot more pathways being measured at the same time. ADME tox for essentially drug therapeutics, and then regenerative medicine and cell-based diagnostics. And that's an interesting little point down there. As a laboratory medicine director, we're just beginning to turn the corner on using human cells as a diagnostic tool to determine an individual's body physiology. And that's an interesting thing that will take cells from your body, put them in the laboratory, and we'll test what systems we think are going wrong, and the cells behaves almost identically to how the cells in the body react to disease. Next slide is talking about cells as reagents, because if you look sort of on the left side of this slide, you start with a stable cell line, then you expand it through bulk cell culture, and then you can cryopreserve a certain amount of it for future use, and then you can reestablish those, obviously thawing them out, and then expand them again. And so usually you use them after several doublings. Or if you want to save time, you can essentially cryopreserve lots and lots of vials and use them after one doubling. So that saves you some time there. And then on the primary cell line, it's pretty much following the bulk cell culture, cryopreservation, and expansion, but there's a movement to dispense frozen cells directly into an incubator and then use them in an assay, or dispense the frozen cells right into the assay and let them warm up and report what they want to report. So the movement is, the industry trend is towards immediate use, and so there's a lot of work going into how to better cryopreserve cells, how to better get them from the freezer into your assay, and how to do that in an automated fashion. Next slide talks about the ideal cell culture method. It should take advantage of the new 3D paradigms, because cells grown in 3D look and behave very much like in vivo, and I'll show you some examples of that coming up. They provide cell morphology that mimics the in vivo counterpart, and then physiologic and biochemical responses are all similar, and it allows this co-culture of cells for this organ model development. The assay or the cell culture method should be automatable to provide convenience and standardization, which are two words that are not often associated with cell culture, but that's where we're headed. We want high yield, and we want to take the art out of cell culture. So there are automated 2D cell culture systems out on the market, the Compact T, the Cell Host, the Cellmate, the CGD Work Cell, and the Sonata. And all of those have seen some degree of success in being able to do bulk cell culture for pharmaceutical companies. They tend to be large, they take up a lot of room, and they don't really perform much more efficiently than people, because they are handling Petri dishes or T75 flasks just the same way a human handles those. And so has been a movement in the automated world to try to essentially redesign it, re-engineer it specifically for automation. And that's what we've done in our laboratory, because we have automated systems and we wanted to get into 3D. So we developed our own for our NIH-funded research program here, and the system has been commercialized and spun off into Charlottesville. So what we have are three things that work together: a bio-wiggler, which is essentially a platform on which you put a 50 mil centrifuge tube, centrifuge-like tube called the levy tube, and the levy tube has little veins in it, and the wiggler does the old washing machine motion of back and forth, sort of 360 degrees in one direction and then immediately reversed to 360 degrees in the other direction. And that wiggling motion can be essentially increased in speed depending on the needs of the cell. So if you want to induce more stress, then you wiggle more. So we get a new term, wiggles per minute is adjustable. And then the third thing is the GEM, which the name of the company was Global Cell Solutions. So it was Global Cell Eukaryotic Microcarriers. So we thought it was a nice way to put it all together and make it sound interesting. So that's, again, invented at UVA to support a cardiovascular research program. So the bench level systems on the next slide are aimed at 3D. The most popular and the one that's been out the longest is the WAVE bioreactor, and that is pictured at the upper right. And what that is essentially a gas permeable bag is filled with media, microcarriers and cells, and it rocks just like a wave. It goes left, right, and all that sloshing gives good oxygenation and nutrients to all the microcarriers supporting cell growth. The Cellagen 310 from New Brunswick is more of a bench-level bioreactor approach, and that has to be taken apart and cleaned after each event. And Corning stepped up to the plate with a disposable spinner flask, so you can essentially put this in your incubator and do standard impeller-based cell suspension kinds of work on microcarriers. The challenge with some of these internal impeller-based systems are that they can beat the cells up pretty badly, and so you have to have a fairly robust attachment of cells to microcarriers that don't essentially get beat up by the impeller. So the 3D matrices that are being used to support cell growth are things like FiberCell, high-clone sphere, microcarrier beads, and then cytopore from Amersham. And so those three in particular, you notice that they're opaque. So it's difficult now that you can see the cells in the high-Q sphere, but that's using a scanning electron micrograph, which few people do routinely. So if you're just taking a representative sample out of your bioreactor or your system and you put it under the scope, you really can't see the details of the cells because they're obscured by that opaque material, usually carbohydrates or plastic. So people have been moving to other kinds of systems. MagCloud and eSci-Bi is various kind of more gel-based systems that give you ability to see the cells, and you can see the spherical shape, and each cell has a fluorescent signature there. So the next slide, just a little question to get you thinking about what gaps I may have in my talk here. Do you currently use this 3D cell culture process in your laboratory? And if the answer is yes, it'd be interesting to know what it is and what results you're getting. So next slide, the advantages of 3D. There are certainly process advantages. Labor, you get about a 75% reduction in labor. Monitoring, you can actually in real time measure cell growth and health. The yield per batch is higher in 3D compared to 2D, and you can do continuous culture, which essentially puts cells into culture and maintain them for months, if not years, under the same lineage. And then the footprint's smaller. 3D takes up less room than standard 2D things. Because, for instance, when we have our I'll show you a picture of our levy tube. It's 50 mils. We only put about 40 mils in there. The amount of cell growth you can get in that is equivalent to 75 T75 flasks. So it's essentially fill 4 incubators with T75 flasks versus just putting cells in a 50 mil tube. The biological advantages of 3D are better access to nutrients and oxygen because everything's being swirled around, better morphology and physiology. You can control stress. Cells is one of the strongest cues that a cell gets is stress kinds of cues. And if you can control that stress, for instance, at the wiggles per minute in our biolevitator, then you can essentially induce different phenotypes that are more relevant. And then certainly you can put more natural growth surfaces into the system, so the cells are sitting down on something that they recognize, and so they tend to say, oh, I'm here, I'm a liver, I need to do this. So the next slide talks about one of the biggest problems in cell culture is that we feed on schedule. So Monday, Wednesday, Friday, you pull off the old media and you put back new media. Sometimes you carry forward some percentage, like 20% of the old media, because it contains the growth factors the cells have secreted. But in any case, you essentially feed it Monday, Wednesday, Friday, and by Monday morning, the cells have been sitting quite a while, and they're getting starving. In this particular slide from an Edward Felder, absolutely no relation to me, but it's I guess all the Felders get into cell culture in the world. But what glucose is the red line, and so glucose is on the x-axis, the y-axis left, lactate is on the y-axis right, and time across the bottom. And you can see that the glucose drops to almost zero, or actually to zero, until the next feeding, and then it's way up again. So those oscillations are kind of like having a sugar fix 3 times a week, and it's probably would lead to cellular diabetes, undoubtedly. And then as the glucose is being metabolized, the cells are giving off lactate, and that gets to a fairly high level. So that up and down kind of thing is not the healthiest thing for a cell, because it's not even close to what happens physiologically. The next slide shows actually what happens at the cellular level to morphology. So here what they did is a static 2D cell culture with feed on schedule versus a perfused cell culture system, because way back then, in 2002, they didn't have 3D cell culture technology available to them. So at the top, in the electron micrograph, if you look at the upper picture, and it says, you see the word basal and apical, there's an epithelial layer, and you can see that there's very few features around. Where if you look at the one on the bottom, it looks much more relevant as a cell, more circular, but notice all of the little microvilli, little finger-like things sitting up there. And that's one of the challenges of 2D biology, is the cell doesn't know how to deploy its microvilli appropriately. And yet in 3D, they can do exactly what they need to do. So enhancing subcellular structures through a biomimetic environment is the next slide, and this talks specifically about our 3D platform versus a traditional 2D platform. So on the left is a transwell, a human renal proximal tubular cell, which is a human kidney cell, or expanding on a filter cup placed inside a well of a microplate. So that transwell is the trade name for that. And you see the black squarish like outline of that cell. Those are gaps between the cells. Now that's not what happens in real life. And also the cells are only expressing 32 microvilli per square micron, and you can see little white dots in that cell, or each microvilli. On the right, that's a picture of the GEM technology, and with cells growing on it. The nuclei are stained in blue with DAPI, and phalloidin stains the plasma membrane. We're currently focused on essentially the most proximal part of that sphere. So we're seeing the cells that are closest to us. You can see that it fuzzes out because the focus is out of plane. But what you can do is just simply grab your focus knob and turn it, and you can optically slice that sphere as many slices as you want, and you can see the sides of cells, and then on the far side of that sphere you can actually see the back side of a cell. So if you'd stain with fluorescent markers for the apical versus basolateral side of an epithelial cell, you could actually see how they display themselves or appear on various aspects of a cell. You can't do anything close to that in the standard 2D world. So then We did some scanning electron micrographs, and you can see 364 microvilli per square micron expressed in these cells, which is over tenfold more than what happens on a transwell. And those microvilli are the transporting members. That's where all the action is. And with these telltale bumps, you see the cell outline, and then there's a big white bump in the middle. That's a common feature of these cells in the human body. And then on the right, if you pull back your focus and don't look so closely, you can see how nicely interdigitated those cells are, and there are not going to be any of the gaps between those cells like you see over there on the transwell on the left. So there are very apparent advantages of moving to 3D biology for relevance. And so here's a very close-up picture on the next slide of those microvilli. And what's an interesting feature in the kidney, particularly in the proximal tubule, which is just after your blood is filtered, each of these cells has a primary cilia, which is those whiskers sticking up in the media there. And so one whisker per cell, and what that is a biosensor. And so that cell is asking the question, okay, the glomerulus has filtered the blood. What's in it, and what do I need to do? If I need to reabsorb a lot of sodium, let me sprout many more microvilli. If I want to suppress sodium because somebody just had a triple Big Mac and a large fries, then those primary cilia will suppress the production of microvilli. So that whole dynamic can't be seen in 2D. In fact, we have a hard time finding microvilli in 2D, but in 3D it's self-evident. So the challenge is migrating to automated 3D cell culture systems, because just going from 2D to 3D, I'll warn you that it is the cues and the way your cells look is completely different. So you have to relearn what to look for in your cell culture system to determine the health and well-being of your cells. So just moving from 2D to 3D, it has a little bit of a learning curve, but then when we migrate to automated systems, it'll be even more of a challenge because of all the programming involved. But as with any technology, it will simplify, and there'll be a bench-level automated 3D cell culture system in not so near future. So looking a little bit more specifically at the GEM technology on the next slide, we wanted to engineer it so it was optically clear, so it didn't interfere with any microscopy. We also engineered it to be non-autofluorescent, so there's zero fluorescence background, so any fluorescence we're measuring is from the fluorescence we add to the experimental system. And then the important thing was we added ferromagnetic particles, so picture little chunks of iron that are about half the size of those little white nuclei, or bluish-white nuclei in that picture. Well, those ferromagnetic particles, there aren't very many in the cell in a microcarrier, but they're enough that we can take a magnet and essentially pull that microcarrier across a slide or to the bottom of a tube to facilitate media change, or just collecting the cells for another use. And here's a picture of the BioWiggler, and I have the orange arrows showing you the back and forth motion, but it's 360 degrees, not 180, which is kind of pictured here. And what's nice is you've got 8 positions on the BioWiggler in which you can do independent cell cultures, each with its own controllable wiggles per minute. And you can control the volume in the tube. So the point of the BioWiggler was to make this fully accessible to an automated system, hovering above it like a robot. So the caps would be removed, the whole thing would be in a sterile environment, and the robot could service the tubes by sampling the media, and then you could change from feeding on schedule to feeding on demand, which is when the glucose drops to a certain point, then go ahead and replenish the media. So the benefits of 3D are the clarity, the non-autofluorescence, its suitability for many, many cell types. We have yet to find a cell that won't grow on a microcarrier, properly engineered with a biomimetic surface. And the wiggle-based suspension is low shear, but you can add more shear by increasing the wiggles per minute. But it does give the cells excellent access to nutrients. And it's suitable for automation because a robot can do everything without having to move items around. This picture on the right is just a gem with ferromagnetic particles, and it was a video that I'm having a hard time getting moving here, but what it does is essentially when you turn on the magnet, the gems will slowly work their way over to one side of the vessel. So the benefits of 3D cell culture on the next slide is they're both biochemical and morphological attributes. There's also enhanced growth of difficult cell lines. There are a number of cells that people find very difficult to grow in 2D. Well, they put them on 3D-like surfaces and they grow readily. You can do on the microcarrier treatments, so pictured are yet some, I forget which kind of cell these are, but lots of cells growing on these microcarriers, and you can electroporate those cells directly on the microcarrier. So you don't have to trypsinize, put them in suspension, now they're very unhappy. Then you electroporate them, and then you hope for the best, where in this case they tend to be a lot happier with close neighbors, and the electroporation process doesn't cause too much damage. There's ease of dispensing them because they can be pipetted in any standard pipette. They can be transfected directly on the microcarrier, and you can cryopreserve them directly on the microcarrier. And I'll show you some data as to why there's some benefits there to doing it. Here's the next slide, the cell culture hypoxia and the biochemical effects. So let's start at the bottom of the slide where I show a petri dish and with our microcarriers in it, and we looked at microcarriers on the bottom of the dish during their growth phase by putting a magnet under the dish and pulling them down to make sure they were fairly well anchored. And on the left, we used a magnet on top of the petri dish to pull them up to the surface where all the oxygen was. And we wanted to prove that there was really hypoxia going on when these were at the bottom of a petri dish versus there was really good access to oxygen when they're all near the top of the petri dish. So the data shows that this is relative fluorescence units on the left axis. We're measuring caveolin-1 secretion, which is a cell membrane structural protein that will get secreted if the cells are having a bad hair day, sorry for the scientific jargon. But so normoxia, you get very low amounts of caveolin secretion, and then under hypoxia you get considerably more. So this led us to believe that really hypoxia is not just standard petri dish cell culture leads to hypoxic conditions. In fact, we have 4 incubators in our cell culture laboratory, and 1 out of the 4 was always giving us excellent cell growth in 2D back in our petri dish days, and the other 3 were not. And we couldn't understand why one incubator, so we moved the cells around to the different incubators, and there was always that incubator that had really good cell growth. And so, a little head scratching, we finally figured out that incubator was leaning against the laminar flow hood, and it was vibrating. So just that vibration, slight subtle vibration of the entire incubator caused the media to stir and fed oxygen to the cells on the bottom of the dish. And so that is was one of the kernels that made us invent this 3D cell culture system. So the effects of nutrients and oxygen control, essentially we wanted to see how the growth rate was in cells on a petri dish versus the microcarrier-based system. And so across the x-axis is the days in culture, and we're measuring cell growth here on the y-axis. And you can see that in the petri dish-based system, you get much lower cell growth. It has that classic S-shaped growth curve where it accelerates, and it begins to accelerate. It reaches a nice slope of how fast it's growing, and then it plateaus off when you reach confluence. Whereas the microcarrier-based cell culture, they get a head start. They grow in a very linear fashion because there's just no impediments to growth, and then it plateaus off, obviously, when the microcarriers get saturated. All surface areas are covered. The next slide shows just a representative number of cell lines that we've tried on our microcarriers: mouse smooth muscle, naturally the human proximal tubule, which is our main research tool, and the ones you might recognize: MDCKs, Actin-93s, KCO2s, CHOs, and on and on and on. The T24T, which is the second one from the left in the bottom row, that's a difficult to grow cell line, and there it seemed it was very happy. It grew well, and we did our studies with it. So other ones are smooth muscle and rhabdomyosarcoma. Those are very difficult cell lines to grow, and Merck put their cells onto our microcarriers, and we're very pleased to see that they wouldn't have any impediments to using those cell lines in the future. The next slide shows the density analysis in the MDCK cell line, Baden-Darby canine kidney cell line. And what we wanted to know is what was the cell density doing on a microcarrier versus a Petri dish. So the left column, the kind of grayed out pictures are on Petri dishes. On the right are the microcarrier-based growth. And you can see we give you the top row is 10x objective and 40x objective on the bottom, so you can see individual cells quite readily. And if you zoom in and just randomly go through the Petri dish, you see about 25 cells per unit area, where on a microcarrier you have 190 cells per unit area. So the question is, why is it so dense on the microcarrier versus the plastic? Well, if you slice that microcarrier and look at those cells, they're growing very columnar shaped, so they dense pack, and that's exactly the way they're seen in the kidney. So the next slide shows the density analysis for Hela cells. And so, again, microcarrier versus Petri dish. And Hela is, the workhorse of the cell culture industry. And we found that you had about 70 cells per viewing field on the microcarrier and about half that on the Petri dish. And again, the dense packing and the columnar thing. The inset's red square with the little dots. So we're essentially counting nuclei per unit area. So we then wanted to look at the expression of a structural protein. So naturally we picked vilin because it's the structural protein for microvilli. If you remember back to that scanning electron micrograph with that field of microvilli and the primary cilia, well, each one of those cilia and microtubules express vilin in large quantity. So we did some Western analysis, and on the Petri dish on the left, we had very little vilin expression, and on the 3D gem technology, we had quite a bit more. So that surprised us that essentially there's that much of a difference. But our pictures bore out that that's exactly what was going on. So here's an example of electroporation on a microcarrier. So conventionally on the right, you do cell culture, and then you trypsinize, centrifuge, and resuspend. You wash, you replate, you incubate, and let the cells recover a little bit, and then you electroporate them and then analyze. Where in 3D, we can just grow them on the microcarriers, wash them, electroporate them right on the microcarrier, and analyze. So it is about a 75% reduction in labor and about a 50% reduction in time. The time of just letting the cells acclimatize is usually the longest part of any process. That's the advantage of automating this process, is that robots don't mind waiting around for things to happen, where people are, they get antsy because they know that they could be more productive if they didn't have to sit and watch cells recover. So here's an example of the electroporated gems with the GFP expression, green fluorescence protein, and each gem is pictured. I have a symbol there, one gem, that you can kind of get the outline of that one microcarrier. And so Hela cells successfully transfected on the gems, and they expressed GFP as shown in the picture, and they stayed on the gems after the transfection, and then the Hela gems were brighter after 24 hours than those grown in traditional culture after 4 days. So that's like a 75% improvement in the electroporation efficiency. And so here's just some quantitative data to show the same thing, but in this case we did T24 difficult to grow cell lines, and there was quite a bit of difference in terms of the electroporation efficiency. The question is, how well did the cells survive following that electroporation event, which is pretty onerous for a cell. Somebody's going to put a high voltage across you and poke holes in your membrane. So they tend not to do well after electroporation. If we look at, again, those difficult to grow lines, T24s, we're looking at percent survival on the y-axis, and in suspension, which is the traditional way of doing it, we get about a 22.5% survival rate, where on T24s on gems, we get almost an 80% survival rate. So we think that close packing and interdigitation of those cells protects them because most of their surface area is spent hanging on to each other, and only one aspect of the cell is out getting zapped by the electric field. We also wanted to look at dispensing gems into a 1536 well plate because that's always a question. A cell is about 20 to 30 microns across, and certainly a lot of them will fit in a 1536 well. But when you now put, let's say, 50 to 100 cells on each gem, how's it going to work in a 1536 base system? So here, Hela cells were used. We examined 6 gems, and there are about 750 cells per gem, and about 5,000 cells for every 4 microliters. The dispensing, and this is Mark who was doing this work, they had a Calypsis instrument with a straight tip dispenser, and they were able to dispense quite nicely. So they checked the heat map of using a cell titer glow assay in terms of how acceptable was the well-to-well variability, and they deemed it acceptable. The cells were cultured for 24 hours, and titer glow confirmation and the cells were still viable after 24 hours. So a 1536 would dramatically improve your productivity, especially if you have 3D technology in your 1536 plate, because it's more efficient for labor and time, and also here in terms of quantity of data producible. Next slide. The cells may be easily harvested if you want to get them off the gems, and what we can do is just use Accutase, and so you see these gems after 5 minutes, they still have integrity. At 10 minutes, upper right, they're beginning to fall apart. At 15 minutes, they're dissolving, and at 20 minutes, they're gone. And because these are alginate-based microcarriers, and they're held together by calcium, so if you just put an EDTA, ethylene diamine tetracyclic acid-like chelator in there, it'll pull out the calcium, and the polymers will just come apart. Next slide shows the survival rates, which are higher on gems versus traditional cryopreservation. So again, we're normalizing to 100% of cells that we froze down, and after cryopreservation in the Petri dish, the orange bars on the left shows about let's say somewhere about 80% survival rate, or let's say it's a vast reduction from 100 down to about 82% or so. But in the microcarrier-based system, you had a much better recovery, up above 90%. So apparently, the cells are protected on that 3D environment. So sorry about the color contrast here, but the Hela cells were checked following the freeze-thaw cycle in a Forsgren dose response assay. And here you've got relative fluorescent units on the left, and the Forsgren log concentration on the x-axis. And you can see that of all the different conditions, frozen cells, fresh cells, frozen cells on gems with 50% confluence, or on gems at 60 or 90% confluent, that the assay results were indistinguishable and not significantly different from each other. So the cells behave appropriately, particularly with Forsgren stimulation. So let's talk a little bit about automated microcarrier-based cell culture systems. When we invented ours, we figured we had a microcarrier, we had a levy tube, and then on the right you can see that picture of a Cavro, I think it is, robot servicing the tubes. And then we have used that basis to put together, and at the lower right picture there is our fully automated cell culture system, still being engineered, but we're close to finished on that one. And what it'll do is essentially it's in an enclosed chamber with appropriately controlled CO2 humidity and temperature, but also oxygen tension. So different cells in the body have different requirements for oxygen tension, and very few people pay attention to that. But here we can essentially administer nitrogen, measure oxygen, and reduce the oxygen concentration through the administration of nitrogen to get to an appropriate oxygen tension for the ideal growth of whatever cell you have of interest. This thing will have 64 independent wiggler spots, so you can grow 64 cell lines simultaneously or random access, on demand, and have the robot maintain them, report to you the health of all of those, or alert you only to challenges and how it's trying to rectify it by feeding more or doing other things, changing the media. And then, essentially you get a little email or text message on your cell phone and say your cells are ready, go on to the next process. This robot is capable of going on to partly onto the next process, but we have some room for microplates so that you could actually take the cells that are happy now, put them in the microplates, add reagents, and go from there manually. But this is our vision of the future. We essentially wanted, so picture there's our automated cell culture system in the middle, but we want high throughput systems, screening automation connected to the automated cell culture system. So those cells could be handed off to that process, which would proceed automatically in the plate reading, and you just get data out the back end of this thing. But at the front end, we were in process of developing an automated minus 150 degrees Celsius cryogenic storage system which could biobank the cells. So the automated cell culture could grow up the cells. Some percentage goes off for assays immediately, and a lot of it gets cryopreserved, which can happen on board here in the automated cell culture system. And then those vials can be handed off to the robot, who would put them away and put them in a cataloged storage. So you could have this thing running 24 hours a day, 365 days a year, generating a whole variety of assays, only limited by your imagination. So that's the conclusion of my presentation. You ought to think about, would you consider the purchase of a fully automated cell culture system for your laboratory? And price is always the issue, and the cell culture automation we're producing is going to be about 1/3 the price of the standard automated systems on the market today. The University of Virginia is looking for an automation partner to acquire this technology and go forth and multiply. So just like cells. So we won't deal with that. So here to summarize and conclude, the desirable outcomes of 3D cell culture is emulation in vivo environment, high cell yields from less lab space, scalable from an individual researcher to larger scale production, significantly reduce cost, consumables, eliminate contamination, reduce the labor. You can easily grow difficult cell lines, automate the entire process, and have real-time monitoring with feedback control. There are issues in transitioning from 2D to 3D. You have to, as I said at the beginning of my talk, I want to reiterate here, you have to learn to recognize new cell phenotypes on these levitated beads, test and use frozen cells and assays. You have to make sure that works in your platform. You have to learn how to manipulate microcarriers and dispensers and plate readers, and look for improved biological responses and improved phenotypes. Corning has addressed the issue of microcarriers in a plate because they have these spheroid plates, or they're essentially slightly concave bottoms on the plate, and all the microcarriers gather in the center of that well and are more readily read by a plate reader or seen under a microscope. So I'd like to acknowledge Merck's tremendous assistance in showing that this actually works in the real world. Not that universities aren't the real world, but there's, where the rubber meets the road is in pharmaceutical industries. We have the group, my co-inventor John Gilday, and a lot of people who spent time perfecting this, particularly Gabriel Smith, who is the main engineer of our cell culture robot. Thank you. So now questions are coming in from the field, and so let's see, what are the benefits of 3D cell culture for stem cells? That's a experiment or a group of experiments we did up at Harvard, And there we showed that putting stem cells on gems maintained them in a de-differentiated state far longer than the 2D-based cell culture systems. So only until you gave them the biological clues to differentiate would they differentiate. So at that point they would maintain on the gems. Now we didn't get a sense of how long you can maintain them, but we've grown our human kidney cells and we've maintained a very stable cell culture for 9 months. And just we draw out a little bit for experiments every day. So there is some benefit in terms of longevity in these systems. Okay, the next question is there significant cost savings on labor with 3D? I think I answered that pretty clearly, but the significance is it's about a 75% reduction in labor, and the labor can be less skilled because if you there's a lot less hand manipulation, which are acquired skills. So if you aren't doing as much hand manipulation, then you can put lower trained people onto a 3D cell culture system and still have the same degree of success. So it'll make cost less on labor per technologist. Another question is, what other phenotype benefits have you seen with the use of 3D cell culture? Well, we haven't really exhausted all of those possibilities, but what we can do is show that the cells not only look better, but they behave very close to the in vivo counterpart. We took some of the renal proximal tubular cells, grew them on gems, but the source of the cells was not the ATCC or a hunk of human kidney, which is our normal source. What the source was human urine, and we have a clinical study going on where we're recruiting 4,000 people into a salt-controlled study where we provide 100% of their consumables over a 2-week period, and then they oscillate from high to low salt or low to high salt diets. And so we wanted to ask the question, how are the kidneys performing? So we do physiologic studies, but then we took these proximal tubular cells from the urine, which are still alive. We cultured them, grew them on gems, and showed that their physiology in response to sodium was identical to the individual it came from. So it's truly personalized medicine. So that's all I see in terms of questions at this point. I'm available. See my email address down in the lower right-hand corner. Feel free to email me with additional questions or information, because I have lots of literature and things to share. Thank you.
Webcast possible. Today's webcast will be available for on-demand until August 2017. Keep an eye out for an email from LabRoots alerting you when this webcast will be available for replay. We encourage you to forward this announcement to your colleagues who may have missed today's live event. Thanks for joining us, and we hope to see you again next time. Bye-bye.